Thank you again for your kind invitation to participate in your meeting. I do hope that after the pandemic, we will have a chance to meet in person. And I wish you well with your meeting this year. Now we're going to consider the role of the oocyte during abnormal fertilization. And again, I would just like to disclose my role as editor of JARG and welcome you to become involved. But also this gives me a chance to share with you a confocal image of a human embryo that fertilized normally, had a very symmetrical cleavage up to the 4-cell stage, and then arrested. Okay, we fixed and examined this embryo sometime after it arrested, but as you can see, it underwent extensive fragmentation. In blue, you can see the genome in some of these blastomeres, if you will. And in others, for example, here we see remnants of what we now understand to be a meiotic spindle. The question we're going to explore in this lecture is, really one about abnormalities in the fertilization process that contribute to poor development of the embryo, but also have probably an origin in the oocyte. And really, as we've alluded to in the previous lecture, there's a lot that must happen in response to the LH surge, okay? Whether we trigger it ourselves in a conventional ART cycle, or whether it occurs normally during during ovulation, it is going to set the stage for the events of fertilization. And in essence, this involves exiting the meiotic cell cycle and entering the mitotic cell cycle. And what happens is the egg, the oocyte, has to be instructed to become a zygote, a role that is attributed in normal fertilization to the entry of the sperm. And there are consequences to both the coordination of the timing from a cell cycle perspective, but also from the perspective of egg activation. Now, what we've come to understand is that the oocyte has to be prepared to support the metabolism, not only consumed during the fertilization process, but afterwards. And that much of the metabolic support for the zygote is derived from the cumulus cells prior to ovulation during the process of oocyte maturation. And this contributes to the zygote, energetic trinucleoside phosphates, okay, in the form of ATP and GTP, which we generally overlook. But importantly, the provision of reduced glutathione, or GSH, which will support development of the male pronucleus. And insights into some of these processes have shed light on the fact that there is a very high incidence of, let's say, genetic plasticity during the first cell cycle or two, that can not only compromise embryo development early on, but it can compromise the health of offspring born from these techniques. And so we'll take a look here. Of course, time-lapse imaging has become a hugely important tool. We often see in very healthy embryos the transient appearance of various blebs and things, okay? And these represent modifications in the cytoskeleton due to a loss of synchrony between chromosome segregation and cytokinesis, or the process of cell contraction. You can see how much motility we're witnessing, okay? And here at a slightly later cleavage stage, okay, we're going to see, you can see nuclei here rotating around, okay? And very often, the division process is initiated, but instead of the cells completing cytokinesis, they will end up with multiple nuclei, okay? And this, of course, is a condition that we can trace back to fertilization and the improper allocation of the organizing principles. There you can see a binucleate cell. Now, we know that many of the embryos that are successfully fertilized and placed in culture undergo these initial divisions and end up with, as I just showed you, multiple nuclei or sometimes smaller nuclei. And the cleavage patterns, which are deduced generally from time-lapse imaging, yield various genotypes when analyzed in specific cases like this. So here the numbers you see, for example, here's one that was unable to initiate the first cleavage, and lo and behold, it has produced 2 nuclei, each containing 92 chromosomes, okay? And you can see the wide distribution of these with or without cleavage that is essentially coming from failure to properly execute the first cell cycle. And you know, this is a particularly interesting example of the events just prior to syngamy. Okay, so if you recall, that dissolution of the nuclear envelope in the male and female nuclei, pronuclei, if these structures are not able to successfully complete the first round of DNA synthesis, then what will happen is when the nuclear envelope breaks down and you need to enter M phase of that first cell cycle, you will get inappropriate localization of these chromosomes, which result in the genotypes that I just showed you. These are very, very common disturbances that are known to occur in human embryos. They were first described in the early 90s by a number of investigators, and they've raised concern now over exactly how capable are human embryos of correcting the mistakes from the earliest stages of development. Now, important to this end is you can see that this is the first polar body here. Here's the second polar body, okay? And that as the pronuclear approximation occurs, what many people have reported is a clearance of the cytoplasm away from the membrane of the zygote. And understand now that, of course, the cortical reaction would have taken place by this point. The zona would be modified. But importantly, this asynchronous migration between the pronuclei is commonly thought to underscore the various genetic abnormalities that we see during early human development. And so, you know, over the course of this, you know, people have been focusing their attention on more and more of these detailed events, again, using time-lapse imaging. And we see that this is a particularly critical stage from the time of fertilization, you know, mostly in cases where sperm entry can be timed by the use of ICSI, to that first cleavage. And the attention has been drawn to, and this has now been seen in other animals too, to this fundamental fact. The meiotic spindle in the oocyte aligns chromosomes without having an astral organization at either of the poles. Whereas the zygotic spindle, and this was recognized some years ago, has at least one astral array of microtubules, and sometimes two. And of course, the textbook that we were led to believe, I'm just going to go back, the textbook understanding that we were led to believe about this is that when you leave meiosis and acquire that male centrosome, its replication would be critical to allowing for the bipolar organization and alignment of chromosomes during that first cleavage division. This is not the case. Okay, this is not the case. Many have now shown that there are a number of maternal factors that have to coordinate with the male centrosome in order to build the bipolar spindle. And as I'm going to show you in a moment, this is a highly suspicious process. That is, the process of syngamy is fraught with errors, especially in the human embryo, and is very likely to be an attribution not so much to the sperm centrosome as much as to the organization of the oocyte cytoplasm and how it is modified during fertilization. We, again, we extensively analyze these interactions. They have a lot to do with establishing that innate asymmetry in the oocyte that occurs during polar body extrusion. And that asymmetry, the punchline is it has to be broken down, okay? A symmetry, an asymmetry breaking event that would allow for axes to be determined in the zygote and the correct assembly of microtubules for that first zygotic spindle. And it turns out there are other manifestations of this. The more common one that's now been observed is you have pronuclear approximation, and then you have cleavage into not a 2-cell embryo, but a 3-cell embryo, okay? You know, as was shown originally by Rubo and others, and now has been very well studied from a genetic perspective, these are attributable to not bipolar but tripolar spindles that effect the segregation of chromosomes in any number of arrays. For example, here would be a 3-cell embryo that might be disomic in 2 of the blastomeres, but nullisomic in the other. And there are various other modifications, including the fact that chromatids don't always split, so you can also have, chromatids from mom or dad that retain anchored to each other, and this, of course, can lead to all sorts of genetic disturbances. Now, the secret behind this is a gene, the polo-like kinase number 4, that has been shown to be genetically mapped to a locus in the female. And this tripolar spindle configuration, again, highlights what I would consider to be a major impact factor from the oocyte in those early divisions. And it does come back to this process of syngamy in that, despite what we all think about the lovely integration of male and female genomes at the time of fertilization, the timing of their proximity and breakdown is critical to assuring appropriate chromosome alignment and equivalent segregation during that first cell cycle. So I think this is one of the more fascinating and interesting things that comes along with the more detailed studies as we have them now here, looking at studies in the mouse, where the distancing between the pronuclei led to irregularities in the spindle shape. In fact, here you can almost see the sense that you have 2 spindles forming. And when these were mapped for Y chromosome distribution, the little spots are the centromeres on each of the chromosomes. It was found that one of these spindles had all the male chromosomes and one of them had all the female chromosomes. So you can imagine here building, a zygotic spindle If you don't have appropriate management of these maternal factors that organize the poles of the spindle, you could very, very likely result in a tripolar, even quadrupolar spindle, which means starting off life. Here's an example of a tripolar spindle. Starting off life with a serious disruption in the segregation of parental genomes. Okay, and this was a study published in JARG a couple years ago, showing the detection of these two discriminating spindles in a human oocyte. So this very much seems like something that affects the human condition, and again, something that would be derived from the maternal side. Okay. And again, the emphasis here has been placed on timing for PN breakdown, which would lead to the asynchrony and the generation of these direct cleavage features that ultimately, in most cases, lead to cleavage arrest and the inability of these embryos to proceed. So again, coming back to my original point, very few of the oocytes that successfully fertilize go on to produce a baby. We're now seeing that interruptions in development after what appears to be successful fertilization have their origins at the time of fertilization in probably both sperm and oocyte contributions. But what I've tried to highlight here are the main contributions that we know are problematic coming from the oocyte. So genetic integrity, as it impacts both meiotic chromosome segregation and that first mitotic division, running out of gas to drive these events. Again, the resources to support these dynamic events after fertilization are maternally derived. It does not seem that the mitochondria of the oocyte are operational at this point to the extent that they could be supporting this. And so how the oocytes matured, their in vivo physiology is essential. We've seen structural abnormalities, and for sure, whether or not apoptosis or autophagy should be activated comes down to whether or not the oocyte has delivered to the zygote the appropriate factors that can make these important decisions, for example, about eliminating cells that may have an abnormal chromosome constitution. You know, as I just said, the majority of these contributing factors that result in abnormal fertilization are oocyte-derived, although certainly the sperm deserves more attention. So we're left with this picture of generation of oocytes, you know, through the stage of gonadotropin independence and dependence, and these final phases of prematuration and then maturation involving key transitions in cell cycle regulation. Certainly, we know that you know, these are more likely to be factors affecting eggs as a function of patient age. And I'm just going to skip through this and conclude there that there are many loci now for abnormal fertilization that we can begin to track back to oocyte quality, which will be the next topic that I will consider this morning or this afternoon with you. Thank you for your attention.